Our next speaker is Henry Mauser. Henry is from the Department of Biological Sciences. The title of Henry's talk is A Microbial Murder Mystery, How the Blood-Brain Barrier Fails During Meningitis. Thank you, Henry.
In a murder mystery, the greatest threat is often the one you least suspect. Pneumococcus, or pneumo, the bacterium depicted in green, is an unassuming microbe. It typically lives in the nasal passages of humans without ever causing disease. You never suspect then that this same microbe accounts for nearly 40,000 deaths per year due to an infection known as meningitis. Meningitis occurs when bacteria in the bloodstream cross into the brain and cause overwhelming inflammation, which often proves fatal. Now, in actuality, very few bacteria are able to get into the brain due to a structure known as the blood-brain barrier. The blood-brain barrier is a network of specialized cells that separates the blood from the brain. You can think of these cells like bricks that form the wall that is the blood-brain barrier. And typically, the barrier is highly effective at keeping dangerous pathogens and toxins outside of the brain. However, we know that pneumococcus is able to breach the blood-brain barrier and access the brain, but how it does so is still largely a mystery. One of the greatest obstacles to solving this mystery is simply our ability to understand this disease. In natural settings, pneumo only infects humans, but scientists clearly can't infect and dissect humans to study meningitis. So we're faced with the dilemma of if we can't study it, we won't understand it. And if we don't understand it, we cannot treat it. My work is addressing this dilemma. My lab and I are able to take non-embryonic stem cells from human donors and over a 14-day process, differentiate them into cells that express all of the key hallmarks of the blood-brain barrier. This cell model has been validated and utilized in numerous publications. I'm not the first to use it, but no one has ever used this model to study pneumococcal meningitis. So that's exactly what I did. You can actually see these cells shown via a technique called immunofluorescent microscopy. This allows us to visualize specific cellular structures. You can see the nuclei of each cell shown in blue, and in red is a protein called ZO1. Now, if the barrier is a wall and the cells are the bricks, then ZO1 is the mortar that holds everything together. It helps create that impenetrable barrier so that nothing can slip through. And it's doing a great job of it in our healthy, uninfected condition. But when I infected with pneumococcus, I observed the loss or degradation of ZO1. This suggests that pneumo is destroying ZO1 as a means of creating a chink in the wall, making a hole in the barrier and slipping into the brain to cause meningitis. This is just one of the many discoveries I've been making, and while the mystery is still far from solved, my work is bringing us closer and closer to our goal of understanding, treating, and eventually beating this disease. Thank you.